First, I want to introduce our seminar speaker today, Dr. Jeremy Claisse, who's at Occidental College. He's currently a lecturer and a research scientist there at the Van Tuna Research Group, working with Dan Pondella. A little bit more about his background. Jeremy did his undergraduate degree at UC Santa Barbara, and then he went to a much more horrible place to live, which was Hawaii, and did his PhD degree there in Hawaii with Jim Parrish. And he was studying yellow tang and looking at their life history and demography, population dynamics, and they're a really important aquarium fishery species. And I think he might tell us a little bit about some of that work today. And then after that, he moved back to California and started as a postdoc at Occidental College, and he's been there ever since, sort of picking up more and more responsibilities for teaching and research and all that. And he's going to be telling us about the work he's been doing there, working in kelp forest systems, looking at productivity of fish communities, and he just had a really excellent paper that came out this week in the Proceedings of the National Academy of Sciences, excellent journal, where he did some of this work looking at productivity on oil platforms. So that's going to be part of the talk today. So I guess, without further ado, Jeremy.
Thanks. All right, so really excited to be here, and I'd like to thank Scott for hosting me and Allison for inviting me. So today I'm going to talk about two different projects that I've done over the last couple years at Occidental, with the general idea being trying to incorporate more biological information, more life history information into producing metrics that then we can use to do ecological studies. So we'll talk about one, calculating fish production, secondary production on oil platforms, and then a pretty different sort of metric looking at gonad biomass production in urchin barrens and looking at the potential of urchin restoration, which you saw a little preview of that video of what that actually means. So I did my dissertation work in Hawaii on yellow tang, and maybe it's a little bit of withdrawal, but it's hard to not do a presentation without showing some yellow tang. Clearly this has nothing to do with oil platforms or urchin barrens, California. But I wanted to give a little bit of background on sort of how I became interested in this approach by telling you just a little bit about the work that I did out there. So this is yellow tang, a sturgeon fish. You're probably familiar if you go to any pet store that has saltwater aquarium fish in it, they'll have these. They pretty much all come from the west coast of the Big Island of Hawaii. Like 80% of the ones in Hawaii come from that coastline, and almost all of them that show up in home aquariums here come from Hawaii. So they're pretty much all coming from that coastline. This is me impersonating an aquarium collector. Basically, we tried to learn their method. So there's a big barrier net. You can see here a monofilament barrier net that's about 3 or 4 meters high. And basically, you just set up the net. The commercial guys use like 3 different nets that they kind of set up. And then you just herd the yellow tang into the net, and they hit the net. Their little snouts get caught in there, and they just freeze. And like, oh, I'm caught. And then you can go up and grab them, and you can actually put them like one in between each 2 fingers, take them back and stick them in this little holding tank. And it's once you start doing it, you realize like why the number one fish caught is yellow tang, because they're very easy. For whatever reason, they just herd right in the net. And we could catch maybe 80 or so in a really good day and tag them and release them. Those guys can go out and catch 500, 600 in a day. So there's a big fishery for yellow tang and other aquarium fish. They started in the 90s. There started to be concerns that fish populations were being impacted by the fishery. After a sort of big political process, right around 2000, they set up this set of protected areas, so these blue areas. And then the state biologists set up, working with Brian Tussaud, who's going to be one of your speakers later in the semester, I think. They set up a series of monitoring sites, so that's all those little dots there. And they started counting fish 6 times a year and looking at impacts. And I showed up in 2002, a couple years later, and they started having questions about, were they really counting the right age class or size class? Were they in the right habitat? because no one had really done the basic life history, habitat use of yellow tang. So that's where my project came in. And I did sort of the whole suite of life history work. We did otolith-based growth studies, mark and recapture studies on movements, site fidelity, habitat use. We found that the recruits and juveniles tended to live in a deeper habitat, and then they moved into a shallower habitat. So you tend to find the adults in the shallower habitat. And that had very important implications for that monitoring data, because they realized that they were just counting the fish in this size class. And there's part of it is that the males and females switch those habitats at different sizes. So, and the males and females look exactly alike, so they couldn't tell. So they thought they were getting most of the size range, but really they were just getting juveniles and maybe some subadults, sort of. So this work, we were able to interpret their data. They added some shallow water transects to start counting the adults, and we were able to better interpret what was going on with the fishery and apply this basic information to understand those counts and those densities. We also did a little reproduction work and found that they spawn on a lunar cycle. It's pretty interesting as well. So the basic life history information can inform interpretation of monitoring data. And now the future of this work is to try to pull all that together into a more formal population dynamics model and do a little bit of fisheries modeling associated with that. But I could really see the value of understanding the species that you're trying to study or you're trying to count by having all this basic life history information. So I finished my PhD and I started working at Occidental College. At the beginning, I thought it was just going to be maybe a 6-month adjunct teaching position, but I've been there 5 years now, teaching classes and working with Dan Pondella and Van Tuna Research Group. We have about 5 professional staff. This is our boat. We can get to pretty much anywhere in the Southern California Bight with that boat. And for a small liberal arts college, we just have like about 2,000 undergraduates. It's a pretty unique situation having this sort of robust field operation. Our lab name comes from a boat that Occidental used to own called the Van Tuna. And if you grew up in Los Angeles, there's a decent chance that in junior high or high school you went out on a field trip on that boat. I went out when I was in freshman in high school. And then in the mid-90s or so, it became too expensive to operate. So they ended up selling the boat. But we still have a really robust operation. We have students doing, we have a scientific diving program. We do a lot of trawl surveys. We do environmental monitoring. So students get a wide variety of research experience in our lab. So the first project I'm going to talk about is work that is built from about 20 years of research that Milton Love at UCSB has done, working with Ann Bull and other collaborators there, going out and studying these oil platforms and the fish living on structures underneath them. And they've been surveying them every year. And then we myself and Dan Pondella largely, took that data to produce a production model I'm going to tell you about. So there's a lot of work that went into collecting the data that made it possible to calculate production in the way that we did. And then other people, a couple of research associates that work in our lab, they did all the literature review to find all the life history parameters that sort of went into this. So secondary production is pretty simple. It's the amount of new biomass or weight of fish produced in an area over some unit of time. So in this model, it's grams of fish per meter squared per year. And you can think of secondary production as a functional measure as opposed to what's referred to as a structure or static measures like density, biomass, even things like diversity, richness. And that's because it represents a pathway of energy energy flow through an ecosystem. So animals eat things, and then they use energy to respire. Some of it makes it all the way through, comes out the other end. They have to protect themselves. They burn it off. And some of it goes into new growth. And in other ecosystems, like freshwater ecosystems, and in some cases marine benthic invertebrate studies, they've been using secondary production for a long time. But for fishes, it's, I would say, relatively underused. We, in the literature review I'm going to show you, we found about 25 studies that produced estimates of secondary production, fish production, for a whole community of fish. And then there's another handful, dozen or so studies where they produced estimates of something that's production related, but we couldn't relate it. They could use them within their study to compare sites, but they weren't available to be able to compare to estimates like we produced or other ecosystems. Okay, so for this study, we wanted to develop a model of production using fisheries-independent data, so basically annual counts of fish and size structure. We then applied that to data that Milton, Love, and others had collected on oil platforms and on natural reefs throughout Southern California. And then we compared our estimates from those two systems, oil platforms and natural reefs, to all the previous estimates we could find for marine systems. So as I've said a number of times already, these are huge structures that Milton's been studying them for a long time, and they are good fish habitats. A number of publications have shown you consistently find a lot of fish living on them. You can see the complexity of these structures, and for a little scale, these are cars down here. So this is one of the larger platforms that is, I think this is San Francisco Bay, that it's going to be towed off and down the coastline at some point 30 or 40 years ago. One of the reasons for studying this at the time we did is, I think 3 or 4 years ago now, California passed a rigs to reef law that may permit some of the platforms to remain in place, or some part of them, as artificial reefs, or remain as artificial reefs. Another reason we wanted to study this is globally there's a big push to produce energy offshore using renewable technologies like wind energy, marine hydrokinetic energy, so tidal and wave energy. So we're thinking that if we can understand what's important about these habitats being good for fish, producing high production, then that might be applicable to some of these new technologies that they're not being put out there to be good habitats for fish, but if we can make them better habitats for fish, then why not? Why not try to get as much conservation or fisheries benefit as you can? So another thing for scaling these platforms, they range dramatically, or a lot in Southern California. The ones in our study are from about 60 meters in depth or tall to about 225 meters. There are some larger ones in California and elsewhere, but these are on the scale of things like the Eiffel Tower, Empire State Building, really big structures. So all the data that went into this is from submersible surveys, so small submarines that Milton and others went down and do transects basically around the platforms or across natural reefs, and they get annual densities of all the fish they see in 5 centimeter size classes. So we have both density and size information. We chose sites that had at least 5 years of data. They had up to 15 years in some cases. So the yellow diamonds are the platforms where we had at least 5 years of data, and they span from about Point Conception. Here's Los Angeles down here. Palos Verdes, the second story I'm going to talk about, based there. Occidental is sort of right back in there. And then the green circles are the natural reefs that we studied, and those two span pretty much the length of that coastline, and they're at a range of depths as well. So we wanted to try to get as good a comparison as we could, given the data we had. to compare production on both these platforms to what's going on natural reefs. So some of Milton's work has shown that on the platforms, you get different communities, and probably more important, you get different size structures of fish living up sort of away from the bottom, up in the structure, up in the water column, as opposed to the bottom 2 meters or so of the platform. And around the base of the platforms, you get a lot of, if you've ever seen pictures of these, all the metal structure is encrusted with invertebrates. Particularly up at the top, you get lots of mussel shells, mussels forming. Those die, fall off. So you get mounds of mussel shells sort of around the bottom. They create these really complex habitats, good habitats for rockfish. So we estimate production sort of separately in this midwater habitat based on the fish observed there, and then also for the bottom habitat. And then what we do is we convert those sort of per meter squared of habitat structure estimates to an estimate that's per meter squared of seafloor. Basically, we add up all the production on the whole platform and then divide it by the area of the footprint of the seafloor, so of the seafloor underneath the platform. So that gives you an estimate that in some ways is more similar to the estimates of production from other ecosystems, largely estuaries. There's a lot of, most of them come from estuary studies, a lot from California and elsewhere, things like on coral reefs, where they're estimated per meter squared of seafloor. It's also, this is another rationale for calculating it this way, is thinking about, well, if they're going to remove the platform, if it's there, you're comparing sort of an all or nothing approach. The natural reefs we looked at, mostly high relief, rocky reefs, sometimes of various sizes of boulders from about 40 to 300 meters deep. But for rocky reefs, they have relatively high amount of habitat structure. Okay, so the model we produced, which we called total fish production, grams per meter squared per year, is the sum of two components. What we called a somatic component and a recruitment production component. The recruitment one is maybe a little bit of a misnomer. It too is somatic production. It's all somatic production, meaning that it's from the growth of biomass or the bodies of the fish. But it's really the source of the data, I guess, that goes into calculating each of these components is why we call it somatic and recruitment. The somatic is, as I'll explain in a second, the fish that are there, and then the recruitment is accounting for all the fish that might recruit into that habitat over a year period, and then how much they grow once they've come into that habitat. Okay, so one of the most challenging things for me writing the paper was figuring out how to do these mathematical notation equations, which took forever using the little math type program. So I thought at least I should show it in the talk. But I'm definitely not going to go through and explain what these are. But it is a good process, I guess, for actually thinking, like really documenting what you have and understanding it. But it's a big pain to actually do it. So the somatic production component, we're calculating over a 1-year period, is made up of two processes. So the fish are growing over that time period, and then there's a survival component. So how much of the fish are going to survive over that 1-year period? And in our model, we only include fish that survive the whole year. In other types of production estimates, like when they work in estuaries, they will go out and catch fish month after month after month and get an average density and how that average density changes over the course of the year. Since our studies were only done at one time period throughout the year, we have to use a survival parameter in our model in order to estimate how much loss there'll be due to mortality in that time period, because we can't directly observe it. So one of the reasons why this work is possible in California is because there's been a lot of life history work done on fishes in this region. So for each species we observed, and we got it for, I guess we did this for about 80% of the species actually in the model and all the important ones. We found within the literature species-specific growth rates, so classic von Bertalanffy growth parameters, as well as weight-length relationships. So we could convert the size of the fish, the length of the fish we do, to a weight. And then we also use these von Bertalanffy growth parameters to plugging it into this model to estimate size-specific mortality. And if you look at these different relationships, so growth is highest, the line is steepest when the fish is small, and then it basically goes to 0 the further out you go. Weight length, the fish are very small, and then it sort of exponentially increases. And then in survivorship, you have very low survivorship for the real small fish, as you would expect. And then survivorship gets higher and higher the larger the fish gets. So these are kind of acting in different, their peaks are at sort of the extremes of each of those relationships. So when you combine them all together, you end up with this relationship. So for the size of fish that we see that's observed, you get a certain amount of production in a year for a fish of a given size, accounting for the amount that are going to survive and the growth rates and the weight-length relationship. So it peaks in intermediate sizes. So very small fish contribute very little to the somatic production component, and very large fish contribute very little. It's just those medium size, those intermediate ages, which are the most productive. So most of the fish on these platforms are rockfish. And another big one, big contributor is lingcod. I'm sure many of you are much more familiar with the biology of these species than I am, given my background. I have to go to Milton a lot to ask him about specific things on the specific species. But when we add up all the production from all the fish living on these platforms, they kind of fall out into two groups. You get the really large-bodied fish, like lingcod and boccaccio, and you can see those here, where relatively few fish can contribute a lot of production. Or you get fish that are relatively small, like square spot or half-banded rockfish, but they are very, very numerous. So they're sort of down in this range. So each individual isn't growing that much, isn't increasing that much, but there's a whole bunch of them on the platforms. Okay, so that's the somatic production component. How much are the fish growing, the surviving fish growing over the course of a year? But we still need to account for recruitment. So that's over a course of a year, you have new fish coming out from their planktonic stage to their more demersal stage, living associated with habitats, hard bottom or hard structure habitats, arriving. And this is a really difficult thing to try to estimate. Ideally, you would want to know how many fish arrive there, and then for any period of time that they're there, any growth that they have, that should be contributing to the production of the habitat. But given we only have one time point to survey them, there's literally tons of these small fish on these platforms. We had to take a very simplistic approach of estimating how much production comes from recruitment. So what we did is the fish that were less than a year old, or we estimate with the von Bertalanffy parameters the size of a fish that would be a year old. We make that the cutoff. And any fish below that size had arrived at the platform in the previous year, grown on the platform, and survived up until the point when we saw them. So we just this is a similar approach that was used by other authors that looked at production in eelgrass beds, I think, or seagrass beds in Japan. So we just take the biomass of those fish and assume that they arrived there, they grew up there over the previous year before we did the surveys. So we get that component as well. And then we add those two together. And we end up with an annual production estimate, so each one of these little circles, for each site, either a complete platform or a natural reef, scaled to grams per meter squared per year. And because we had to deal with a few issues that not every platform or reef was surveyed every year, and year to year there's big differences in recruitment, fish recruitment. So we had to account for the site and year factor using a linear mixed model analysis. So basically you have your fixed factor that typically the factor you mostly care about, in this case, production on natural reefs versus production on oil platforms. And then you have these other factors which you want to try to account for in your statistical model. So we use that approach, and we end up with, across all platforms, an average of 189 grams per meter squared per year being produced, compared to 7 grams per meter squared produced on the natural habitat. So this is about 27 times higher when you compare them. And this is not, it was, well, I'll show you the individual metrics, individual platform metrics in a second, but this was not that surprising considering how big these structures are and how many fish we knew were living on them consistently based on Milton's work. But the magnitude of these was surprising when we compare it to other studies. So we can do the same thing across individual platforms. So here are the individual natural reefs that we looked at, and then here are the individual platforms. And this is broken up in the recruitment production component, so the amount of production from the fish that arrived during that year, and then the somatic production component, or the amount of fish that grew, that we observed and then grew over a year. And again, you can see there's a lot of variation platform to platform, and then there's a lot of year-to-year variation, so that's the big error bars, based on high variability in recruitment. So one thing that's kind of important that we took from this work is that you do need a good amount of data to make estimates like this. You need repeated, we sort of set an arbitrary cutoff of 5 years, but you need many years of data in order to be able to get even somewhat precise estimates because it's just going to vary so much year to year based on the recruitment over the previous few years. So we did a literature review of all the production estimates that we could find for complete communities of fish. Most of the ones we found were from estuary habitats, a few from here in California. Larry Allen did his PhD. He's at Northridge down by us. We work with him on a number of projects back in the early 80s in Newport Bay down in LA. And at the time when he did his work, this was the highest estimate that was available. So these range from about 3 to about 75 grams per meter squared per year. The highest ones are from estuary habitats, and there was one so far done on a coral reef, and they had a very high estimate, 75 grams per meter squared per year. This one did use a more similar approach to what we did, taking the observed fish and then using life history parameters to calculate it. But again, doing this in coral reefs where the fish communities are very diverse and there haven't been as much life history work done on those fish, so you don't have the whole suite of fish that you can know the growth rates of, sort of prevents this approach being used on a lot of coral reefs currently. You need an area like California where you have a lot of life history work being done previously. So here's if we put those values, the literature values, against ours for the complete platforms. And the platforms are about an order of magnitude higher. So they range from about around 100 to 800 or so. So calculating it this way, these are the highest production estimates that have been calculated for marine systems. I would guess if we're able to do production rates on more coral reefs and some other ecosystems, we will get others that are as high as this, or definitely higher than the highest so far. But this was pretty surprising, how large these estimates really are. So one of the things we want to do next is ask why are some of these higher than the other? And if you just, one thing you might think is it's just based on the size of the platform. The larger it is, the more production. But that's actually a very loose correlation. There's other factors going on, like the location of it, probably in the local oceanography, as well as something about the structure itself that is resulting in this variability in production rates. So that's one of the things we want to do next, is start to ask why are some platforms higher than others. That's the first thing there. And a big application of that is what specific elements could we take from these platforms and then apply to new structures that are being put out there in order to make those be better habitats for fishes. We also need to look at, so far we know that in each one of these little spots where the platforms are relatively small spots, maybe half of a football field sort of size is what these bases of these platforms are like. They're very high productivity, but in a regional scale, if we compare it to all the production on all the natural reefs in the region, are they producing a very small percentage or something that is more sizable? Milton's done some studies, like looking at Boccaccio and just looking at like the numbers of recruits in these habitats compared to natural habitats. And he showed that for an overfished species like that they do contain a relatively high number and are probably important in rebuilding the fishery for that species. But we really do need to understand from a broader perspective how much of the regional production they're contributing. And then we're working and collaborating with Scott and Heather on trying to get the whole suite of life history characteristics for kelp forest fishes, so we can apply this model to kelp forests to be able to look at production under different circumstances in that ecosystem. This work was funded by BOEM, which used to be MMS before the oil spill in the Gulf. So they're clearly very interested in understanding the productivity of these structures. So I'll move on from there. So now I'm going to talk about a different circumstance or a different project where we're going to look at the amount of gonad in urchins and how that potentially relates to kelp forest habitat restoration, which I'll explain what that is in a little bit. You saw a little preview of that in the video. In a small area along Palos Verdes, or along the Palos Verdes Peninsula, where we do a lot of work. So here's Southern California Bight again. Occidental College is over here. And this is going to focus right along the Palos Verdes Peninsula, so basically surrounded by Los Angeles, LA Harbor's over here. So the largest population center on the West Coast is right around this area. So it's heavily impacted by people. So as I'm sure most of you are familiar with, probably a lot more so than me, macrocystis is the dominant biotic habitat forming component of kelp forest in Southern California, and I think up through here, at least this part of California as well. Supports hundreds of species, both from feeding on kelp as well as using it as habitat for finding food and hiding from predators. The red sea urchin is one of the largest fisheries in California. Typically, it's in the 4, 5, 6 ranking for both weight of animals taken as well as in overall value. And it's harvested and sold for uni, or the gonads of the urchins. Who's eaten uni before? Who likes uni? It's pretty good. The one time my wife tried it, she got sick, and then she's like, I'm never eating uni again. But it's a huge fishery. It's sold both in restaurants here in California, as well as a lot of it shipped to Japan. In the region of California that we're in, particularly around Palos Verdes, Malibu, you have these vast expanses of reef where it's heavily dominated by purple urchins, so a different species of urchin. They're much smaller. They're not fished, primarily because of their overall size. They can't get enough gonad out of them to make it economically viable. You do see some of the red urchins here, but the numbers in our region, they're somewhere around like 90, 98% in that range, depending on where you are purple urchins that are in these barrens and causing these barrens. They're basically overgrazing and eating all the macroalgae in the area. So it's also well known that urchin predators have declined. In our region, there haven't been sea otters for over 100 years. So the primary predators down there on urchins are California spiny lobster and sheephead. Scott's done a lot of work on these. So most likely that is a major factor resulting in urchin barrens, in our region at least, is to some degree overfishing these predators and reduced abundances of these predators. There's been a number of studies, there's been a number of studies in Southern California that have sort of hinted at that or had some evidence show that. In some of Nick Shear's work in New Zealand, they've done work where they have looked at marine protected areas over time and seen how when you bring back the predators, they feed on the urchins, and eventually you get the kelp coming back. But that process takes typically about 10 to 15 years to build up large enough predators so that they can feed on the urchins, and then having that effect to have the kelp grow back. So about 15 years ago, an NGO started doing some restoration projects, basically going out and removing urchins from barrens along Palos Verdes and along Malibu. And they showed that if you remove the urchins, and in most cases, 98% of the urchins they removed were purple urchins in this case, within about 6 months, you get the kelp coming back. And they were in the process of trying to get permits to expand this across Palos Verdes. So that was sort of the stage at when we went and did this study. So one of the things we did is we went out and mapped the urchin barrens along Palos Verdes, and that's in the red here. And we found there was about 61 hectares of urchins, primarily in this region. Concurrently, the new MPAs across Southern California were implemented in January of 2012, and they brought in they implemented two MPAs. They're basically the same thing. This one has a few special rules associated with it, but they're both basically marine reserves in this region as well. So overall, we wanted to ask, well, if what the fishermen are after is gonad tissue or gonad biomass, you need to have kelp to produce gonad biomass. In barrens, the urchins do not produce large gonads, so they basically don't fish for urchins in barrens. How much potential benefit could you have if you can turn urchin barrens into kelp forest? And kind of a secondary question was, okay, the urchin fishermen are losing this much habitat. If we were able to restore all this habitat that's urchin barren to kelp forest, would that be able to offset potentially the habitat that they are losing here in terms of the amount of gonad biomass available to the fishery? So if you look in urchin barrens, even with red urchins, even though the scale for purple urchins is they're about at 1,500 to 2,000, per 100 square meters in these habitats. But you tend to have about 5 times as many red urchins in barrens compared to those in kelp forests. So in the next few graphs, all of the red is sampling we did in urchin barrens along this region, and then the green is in kelp forests. So you have this one factor where there's more red urchins in barrens, but those urchins are smaller compared to kelp forests. So you have those two factors to deal with, as well as urchins eat kelp. So we went out and looked at the gonad production in kelp forests versus barrens, and it's clear just looking at this picture. In urchin barrens, urchins have much larger gonads than those in kelp forests, and we created a relationship there. And you can see both the size structure issue, where the urchins tend to be smaller in barrens than they are in kelp forests, but the gonad produced, the weight of gonad produced is much higher for urchins in barrens or in kelp forests than in barrens. So we have these 3 different factors, and how do we combine these 3 different factors together? That was sort of the initial problem that we were dealing with. So we used a Monte Carlo simulation to be able to synthesize both sort of the mean value and the variability around that in all 3 of these parameters and combine them together into a single estimate or single metric of the total gonad biomass available to the fishery. So I'm going to walk you through briefly what a Monte Carlo simulation is, in case you're not familiar with it. And it's really, the math is relatively simple. You just need something like R or MATLAB to do it for you and run it, repeat it over and over again. So what a Monte Carlo simulation is you take your empirical data. So we went out and estimated the abundance of urchins per transect. We went out and counted urchins on transect. And then you estimate the probability distribution based on that empirical data. In this case, we used a negative binomial distribution. And this is something that is very applicable to things like counts of fish or invertebrates on a transect, because you tend to have, particularly when they tend to be very clumped. So you tend to have relatively few transects where there's a lot of them, but you have a lot of transects with 0s or 1s or 2 urchins on your transects. So we estimate this probability distribution. We then go and randomly sample from this distribution, so according to the percentages in that, and in R, that's really easy. There's a function that does that for you. So we'll go and sample out, let's say, 5 urchins per 1 transect, and the next one we go and sample, and it's a 0, and the next one it's a 2, and we keep doing that until the area of our transects equals the area that we're interested in. In this case, for the scenario I'm going to walk you through, it's trying to estimate if we could convert all of the barrens into kelp forests, how much gonad biomass would we have? So we randomly pull out abundances, and we add those all up until the total area that we've sampled from all those transects, equals the area of all the urchin barrens outside of the MPAs. So in that case, 36 hectares. We add all those up, and we get an estimate of urchins in all of those habitats, all that habitat. So we have some number, let's say it's 10,000. Then for each urchin, the simulation goes through and first it randomly picks out a size, again, based on the size, the probability distribution of the histogram, basically, of sizes from empirical sampling that we went out and did. So in this case, let's say it'll randomly pull it out and tell us the urchin in our simulation is 100 millimeters. If it's larger than the minimum legal size limit, so urchins in California red urchins have to be 84 millimeters in order for the fisherman to be able to take them. So if it's smaller than that, like if it had given us a 60 millimeter urchin, we would just stop there and it would get a 0 value. If it's larger than that, then we take that size and we go to our next probability distribution, in this case, the relationship between size and gonad weight, and we look up from 100 millimeters to where that is on that curve. Again, that has variability associated with it. And then we randomly draw a gonad weight from that according to that distribution. So it walks through and incorporates all these different types of relationships and variability associated with those relationships. And then we add up the gonad. We do this for all the urchins in our 10,000, or R does this for every urchin. We add those all up, and we get one value. So we might get that there's 3,000 kilograms of urchin biomass in all that habitat. And then we run that simulation 1,000 times. Takes about 25 minutes or so to do all of those. And it gives us a distribution of total gonad biomass weights in all of that habitat. Then we take the 95% confidence interval. Basically, we look at where 2.5% of the samples were below the lowest 2.5, and we sort of eliminate those. And then we look at where 97.5% of our samples were in, and we eliminate the ones above, and we take these lines as our 95% confidence interval. And then we produce one for how much biomass is there currently in all of the urchin barrens, and then how much biomass would we have if we convert it to kelp forest. We assume that we can convert it into kelp forest. And we end up with about a 900% or 9 times increase in gonad biomass available to the fishery if we are able to convert all the barrens, this is per unit area that we're able to convert from barren to kelp forest. Then we did the same thing for getting the total amount of biomass available to the fishery in the barrens, if we restored all of the barrens to kelp forest, and then we also looked at how much is currently available in the MPAs. So how much is technically being lost to the fishery, that no longer available to them. And if we can restore all the kelp forests, all the barrens to kelp forest, it would offset about 50% of how much is lost to them. So a pretty sizable amount. So this work helped contribute to understanding that there is some potential for this to actually provide benefit to the fishery. The fishermen were really interested in that, and they actually went from kind of being against the restoration efforts to being more for them, and now they're actually participating in them. They're actually getting paid to participate in them, so that's a big benefit to them, but they are by far the best at this. And okay, so I'm going to show you the music you heard earlier is a video that one of my undergraduate students made when she went out with her GoPro and participated in restoration, which is basically just going out there with a geological hammer and killing all the urchins in the urchin barrens. All right, so you get the idea. So originally, when they did these pilot studies, they were actually taking all the urchins and relocating them, basically dumping them offshore. But this is what Department of Fish and Wildlife came up with as the best solution for this, thinking that when they kill the urchins, even though they don't have much, the urchins in barrens have very small gonads, they don't have that much nutritional value, there's still something there. So then that will help contribute to the local fishes. And you see, when you start smashing urchins, you just get these huge schools of fish following you around. And then if you come back there, like we would go and try to survey a couple months later, you just get all these fish swimming up to you when you go into these restoration areas. So this was done about a year and a half ago. And now they started basically up in this area, and they're kind of working their way south. And we've started to go back and look at, did what we predict actually happen? Do you see how fast does the kelp come back? Do the urchin start producing large gonads when that kelp comes back? And we've seen both. Within about 2 months, you start to see a lot of sort of the smaller turfs and macroalgae. And then within 4 months or so, you have all this young macrocystis growing up and around. So now we're going back in to those areas that they've restored and looking at how the invertebrate communities have changed. How is the size structure of urchins changing in these habitats, to try to get a more complete view of how it's impacting the system. And I think this whole Palos Verdes region is going to, over the next 10 years or so, be a pretty interesting case study in ecosystem-based management. So the restoration efforts themselves, they're actually applying community ecology to try to do something positive, impact both the overall system by having more kelp be there and all it provides, as well as providing a benefit to the fishermen. And we also have these 2 MPAs where restoration is not occurring. So we'll see, as the predators start to come back, do we see the urchin barrens decreasing in prevalence in those areas? And over time, we can compare the difference between those areas. We can see if the restored areas are maintained, or if without the predators there in these outer areas, if they eventually do come back. The pilot studies they did, those have remained kelp forest sites both in PV and Malibu for about 4 and 10 years now in each case. But it's kind of an interesting case study of trying to do something like this restoration that might mitigate some of the socioeconomic impact on the fishermen who lost habitat, lost area that they're able to fish, by sort of combining these 2 different methods in a more sort of active management approach. So this work, we've had a lot of different funding for different parts of the project, in the monitoring and doing the urchin gonad study. And we had a lot of different undergrads that work with us and our partners at different nonprofits in the area. And with that, I'll take any questions. Yeah.
So Jeremy, you produced a pretty good estimate, in my opinion, of fish production of the Murex compared to reefs and of urchin production in kelp forest. You put it down to fish production, and they were 3-dimensional habitat like kelp forest.
So, no. But, so one of the criticisms, I guess, from the reviewers that we got on the paper we just published is that you're comparing apples and oranges because you have these big structures. And in some ways, yes, these are unique, different habitats. Lots of surface area of hard substrate in a relatively small area of seafloor. And that's a big reason why they're very productive. On the other hand, when, like in estuary habitats, they do sample the fish from the seafloor to the surface. They go out and do beach stains, trawls, perch stains, and they try to get all the fish living from the bottom to the surface. So in some ways it is comparable. You just have these very different reef structures that create habitat for recruits up in the water column, as well as for the older fish as they kind of move down the platform towards the base. So you do provide habitat for fish sort of across those different life stages. But that's another reason why we want to do this work in kelp forests, to see how much we get if we're able to integrate across things that are 30 meters tall and incorporate all of that, all those fish in there.
So is that underway?
Yes, we're in the process of trying to compile all the life history for the kelp forest species. And then it's just a matter of adapting the code over there. Yeah.
So along those lines, I was thinking, let's say you had an isolated natural rocky creek, right, that blows up off the rock. I don't think anybody would calculate production, there's no projection of that reef on the bottom. They don't connect that around it and do it per square meter on the reef, right? So what kind of number do you get if you take the surface area of the platform itself and divide that into the production?
That's a good question. So, that's another thing we talked about in papers. I didn't talk to you about time on this. So this is another model. Looks like the other one, but this is production per meter squared of mid-water habitat, per meter squared of habitat on the base, and then this is natural reefs per meter squared. So maybe in some ways, if you think about demersal fishes, you want to think just about those living right around the structure. And they're, particularly at the base habitat, they're clearly higher. And we do get some values for some platforms, some years, that are in just that relatively small volume, right, than 2 meters of the platform structure, that are as high or higher than those in other ecosystems. And we can also look, this is kind of a complicated plot, but basically this is the recruitment production component, somatic production, sort of split in both the base habitat is the white and the gray is the midwater habitats for individual platforms. And we do see individual platform averages that are in just the somatic component itself that are on par with those from other really high productive ecosystems. So per meter squared of platform structure, we get really high estimates. And then when you multiply those across this big structure, you get enormously high estimates. And we knew that these estimates were going to be high, so we tried to do a number of things in our model to make it more conservative. We didn't include anything that wasn't a demersal fish or something that's going to be more resident. We excluded all the highly local fish. So if schools of anchovies or something that weren't going to reside on that property for a long time were observed, we didn't include them in the model. And the model actually only includes fish living within 2 meters, basically, of the platform structure. And there's huge volumes of water underneath, in that empty space underneath the platform, of fishes that are that weren't accounted for in this model. Particularly, Milton said on years when high recruitment years, you go and it's just like packed with recruits as far as you can see. So the high years are probably way higher than we estimated. So the variability is probably even higher, but we just wanted to keep it as conservative as we could, I guess. Yeah, so.
Another couple summers there when Milton's group were going out diving, right, and they were finding pinnacles, kind of deeper pinnacles that were diveable, that were sort of analogous in a sense to sort of these other platforms.
Yeah.
So did you use any of those for those calculations of natural resources? You could sort of calculate production on this huge pinnacle for a certain area of bottom as being very analogous to
we didn't because we wanted that 5-year cutoff for each site, basically. There is there have been Milton's group, Chris Lowe's group has done work on the tops of platforms and scuba diving depths. And there are applications of that information that we sort of use to interpret our results. One thing about, so if they are going to do the deconditioning process where they allow them to leave the base of the platforms in place, they're going to chop off the top 85 feet. So they'll start 85 feet down and then go down from there. And one of the questions is, well, if you take off that top part, are the recruits still going to find it, or is that going to be an issue recruitment-wise? And so that was one of the reasons why Milton went out and surveyed all those pinnacles to see, do you still see relatively high levels of recruitment around those pinnacles? And you do. So that shouldn't be an issue, I guess. Another thing, so some of Chris Lowe's students' work has looked at the species living at the top 30 meters. And we actually, in those data, there's some that are up about 30 or 10 or 15 meters deep, or kind of the shallowest surveys they do. But really, we don't sample the upper portion that well. So particularly for the southern platforms, we're not including as much production from things like chromis or like compass and Garibaldi, that sort of stuff that's going to reside at the top. But for the deeper platforms, that's a relatively small percentage of the total amount, so we didn't think it was that important. Yeah.
Did you standardize any of those platforms by their depth?
No, but we did do, and so we have kind of another paper that we've written, we're in the process of submitting, where we looked at correlations between production on platforms by depth, and there's not, and so depth and size are highly correlated with each other. So you can look at one or the other, but it's a relatively weak correlation between the size of the platforms or the depth they're in and the overall amount of production. But taking a closer look at platform by platform and comparing platforms that have similar depths and looking at why one may be more productive than the other, that's what we need to do, I think, to sort of better understand the system move forward from there.
Do you think the production at platforms versus the natural reefs is driven by different species? I mean, it looks like you get millions of gotcha on all these platforms. You see the same kind of species combinations.
I don't have that slide. They are similar, but there are some differences in the relative abundances of different species. And like one of the big things we think that's driving the high production on these platforms is having the structure going all the way up to the surface, and having that structure where up, the top half or so, or top 2/3, you see a lot of recruits and a lot of young fish. And then as they get older, they kind of move deeper. So having that structure up there does lead to much higher recruitment rates, and when you compare the recruitment production component, it's much higher in the midwater habitats compared to either the base of the platforms or the natural habitats. Yeah.
Last question.
So in that area situation, so you've got about, again, you estimate about 68 meters of most area. So has anybody done a calculation based on your data to say, okay, well, if you did convert all that calculator, and assuming that X amount of the fishes would be harvested, how much money would the fishermen make versus how much money is being spent to do it?
That's a really good question. I think,
It's kind of mostly political, yeah.
yeah, I mean, I think that's a really good question. No one's, I don't know if the NGOs want to ask that question, like how much are we spending, but I think the way that we tend to look at it is that, so they're definitely spending a lot of money to organize the volunteers and pay the fishermen to go out and do the restoration work. There's also there's another part of the peninsula, there's big sedimentation problems with the cliffs basically collapsing, and where Donald Trump overwatered his golf course and you had a big landslide at Trump National there. There's a big sedimentation problem. So now we're working with NOAA on a project to put artificial reefs in there, basically to build up sort of put these high relief reefs to get above the sediment as another attempt to try to increase the fishery resources along this stretch of coastline. And I should say that the money that comes from this is actually from the Montrose Settlement Restoration Program, MSRP. It's a NOAA program, basically from chemical companies dumping DDT and all sorts of stuff in the LA River back 60 years ago or so. They had this huge settlement, 20 years ago, that created this grant to NOAA that has all this money to try to restore habitat or increase they work on birds, they work on all sorts of stuff to try to get some sort of restoration benefit to account for all the DDT that's basically out there in the southern canal. So they have this big chunk of money, this is what they're deciding to do with it, and trying to make the most of it, I guess.
So they're not considering any kind of aquaculture for sea urchins?
No, not currently. I think there's a lot of work that goes on in Japan that
I know
does this.
there's some people in Norway too that actually are very successful at it.
Okay, yeah, in our region, it's not something.
It's too bad, because I mean, it'd be pretty cool.
Yeah, oh, totally. And there's a group from Japan actually over that went out with the NGO that's doing the restoration work just this past week. Because in Japan, they do all sorts of stuff where they have all these urchins on reefs that are essentially barrens, and then they bring in kelp and feed on the kelp and then harvest them. So they do all sorts of stuff to try to make highest quality urchin feeds possible.
All right, let's thank our speaker.